Hi, friend. I'm so glad you're here. I'm studying for my bio final, but please keep me company. Help me remember everything from class. I like to repeat words and test myself. Okay, let's focus and study. Mitochondria, mitochondria, mitochondria. Centrosome, centrosome, centrosome. Polypeptides, polypeptides, polypeptides. Cardiolipin, cardiolipin. Citric acid cycle, citric acid cycle. Glycolysis, glycolysis. Carbosylation, carbosylation. Mitochondrial matrix, mitochondrial matrix. Cellular proliferation regulation, cellular proliferation regulation. Calcium signaling, calcium signaling. Metabolic reactivity, metabolic reactivity. Okay, I'm pausing here because this section, I'll have to just read it out loud. It's very important. Try to remember this part. I know we'll need this one later for the exam. Okay, so it says recent advances in the identification of the tethers between the mitochondrial and endoplasmic reticulum membranes suggest that the scaffolding function of the molecular elements involved is secondary to other non-structural functions. Fascinating, right? I can read it again. Actually, it would help me too. Okay, here it is. Recent advances in the identification of the tethers between the mitochondrial and endoplasmic reticulum membranes suggest that the scaffolding function of the molecular elements involved is secondary to other non-structural functions. Okay, I think I got it. Let's go back to the key terms here. Endosymbiotic theory. Endosymbiotic theory. Prokaryotic cells. Prokaryotic cells. These cells were the ones hypothesized to be capable of implementing oxidative mechanisms that were not possible for eukaryotic cells. Okay, highlighting that. Okay, redox proteins. Redox proteins. CORR hypothesis. CORR hypothesis. Alpha proteobacteria. Alpha proteobacteria. Electron micrographs. Electron micrographs. These first appeared in 1952 and were the first high-resolution way to visualize mitochondria. Okay, so this next chapter was about plastids, the membrane-bound organelle found in the cells of plants, algae, and some other eukaryotic organisms. Plastid, plastid, intracellular, endosymbiotic, cyanobacteria, photosynthetic pigments, photosynthetic pigments, phycobilin pigments, phycobilin pigments, oxygenetic photosynthesis, oxygenetic photosynthesis, Biosynthetic pathways. Biosynthetic pathways. Chloroplast envelope. Chloroplast envelope. Bryum capillary. Bryum capillary. Kleptoplastidae. Kleptoplastidae. Plastoglobuli. Plastoglobuli. Wow, that's a fun one to say. My notes here say they're found in all chloroplasts and become more common when chloroplasts are under oxidative stress. They contain both structural proteins and enzymes involved in lipid synthesis and metabolism, and contain many types of lipids including plastoquinone, vitamin E, carotenoids, and chlorophylls. Okay, I'm highlighting that. Okay, rubisco. Rubisco. An enzyme that fixes CO2 into sugar molecules. In C3 plants, rubisco is abundant in all chloroplasts. Photophosphorylation. Photophosphorylation. Photosystems. Photosystems. Electron transport chain. Electron transport chain. Adenosine. Adenosine. Photorespiration. Photorespiration. This one occurs when the oxygen concentration is too high and rubisco can't distinguish between oxygen and carbon dioxide very well. Okay, I think that one we should highlight. Okay. Biological containment strategy. Light-dependent reactions. These are the chemical reactions that convert carbon dioxide and hydrogen carrier compounds into glucose. This section here was about distributions within the plant. Okay, cacti have chloroplasts in the stems, but most plants concentrate their chloroplast in the leaves. Let's go ahead and highlight that too. In some algae, mosses, ferns, and flowering plants, chloroplast movement is influenced by red light as well as blue light. Blue light generally causes chloroplasts to seek shelter, while red light draws them out to maximize light absorption. This is all really great information for the exam. Okay, going over to some more key terms here. Hypersensitive response. Systemic acquired resistance. Salicylic acid. Hey, I put that on my face. Jasmonic acid. Nitric oxide. Reactive oxygen species. Gene expression. Gene expression. Photosystem 1 and photosystem 2. Ribulose. Biphosphate. Carbonic acid. Carbonic acid. Nucleotides. Nucleotides. Amyloplasts. Amyloplasts. Plastid dividing rings. Plastid dividing rings. Okay, this next small section was about photosynthesis, specifically, so some terms from here. Alright, carbon concentrating mechanisms. Oh, I know that one. Okay, carbon concentrating mechanisms. Crassulian acid metabolism. Calcium oxalate. Calcium oxalate. Side note, this is a calcium salt of oxalate. All forms are colorless or white. Probably not that important for the exam, but I thought that was a cool fact. Okay, next here, Amaranthus hybridas. Dynamic carbon pools. Dynamic carbon pools. Chlorophyll fluorescence. Chlorophyll fluorescence. Chlorophyll fluorometers. Some of these are hard to say. Okay, let me try that one again. Chlorophyll fluorometers. Okay, yeah, I remember the professor saying that one in class and he stumbled too. Okay, quantum walk. Quantum walk. Let's review this one because I think we need more attention on this. It's a little tricky, at least for me. Okay, so it's a phenomenon that increases the efficiency of the energy transport of light significantly. In the photosynthetic cell of an algae, bacterium, or plant, light-sensitive molecules, chromophores, are arranged in an antenna-shaped structure called a photocomplex. Okay, that's a good refresher for quantum walk. The factors that affect photosynthesis are light irradiance and wavelength, carbon dioxide concentration, and temperature. Total photosynthesis is limited by a range of environmental factors, including total amount of light available, the amount of leaf area available to capture light, the availability of water, and suitable temperature. Okay, and then finally here, these are some more terms that will be in the exam that we should freshen up on. They're not as important, but we still need to review, I think. Okay, phosphoglucolate, integrated fluorometer, fluorometer. Oh, I think that one was in our notes before, I think. Reviewed that one. Okay, daily light integral, elaioplasts, chromoplasts, kilobase blastome, Okay, I think that's all the terms for this final section. I'm honestly really looking forward to acing this exam. We reviewed all, I think, 34 concepts, and I think we have a really great grasp of all of them and a good understanding. The professor will be really proud of me, and you for helping me remember all of them. Thank you, friend. I hope to see you next time.